Welcome to another screencast where this time we'll be addressing the regulation of gene expression in prokaryotes, but we'll be talking about the trp Operon, which is a repressible operon. Okay, so by the end of this, well, you should already be able to do this, right, from watching previous screencasts. The parts of the operon, the regulatory gene, and the repressor protein. So being able to describe what's the relationship between these things here, and then what's the relationship between these guys, okay? And then by the end of this, you should be able to describe the functioning of the trp Operon and explain why the trp Operon is said to be a repressible operon. And then finally, at the end, you should be able to go back and then look at the similarities and the differences between the trp Operon and the lac Operon in terms of regulation. So here we go again, prokaryotic cells transcribing and translating sort of simultaneously, or as soon as the transcript comes off, the translation is going to be started immediately, and so the ribosomes get onto that mRNA are then transcribing it. So again, we are dealing here with prokaryotic cells, and other screencasts will deal with the eukaryotic cells. So regulation in prokaryotes here, the operon. This repressible operon is the trp Operon, so it's similar to but different than the lac Operon. So let's point out over here, here is our regulatory gene. This one is trpR, so when trpR is transcribed and then the mRNA is translated, you get this inactive repressor protein. So this is the default. So the regulatory gene here, the expression of that yields us an inactive repressor protein, which will then, of course, not be binding to the operator. So if that repressor protein is not bound to the operator, then our RNA polymerase is allowed to bind to this promoter, right? And the RNA polymerase can then move down the move through the promoter, move down the genes, the structural genes of the operon, right? And transcribe these genes. So in this case, what we're showing you as an example of this are 5 genes, A, B, C, D, and E, that when they are transcribed, you get this mRNA transcript here, and there's an mRNA transcript there, and so on and so forth. And in prokaryotes, these transcripts are all linked to one next to each other. But the result then of the translation of these transcripts, right, all linked together, are polypeptides that make up enzymes for tryptophan synthesis, right? So the cell, for this example, tryptophan is being produced, and there's a 5-step process that makes tryptophan for the cell. This tryptophan, incidentally, is an amino acid that the cell can synthesize from precursor molecules. So, but it needs enzymes to do that. So if you're making one enzyme, you make them all, and therefore you end up getting tryptophan. Okay. So as it sits right here, this operon is on, but can be turned off, hence it's then repressible. So how do we turn this off? Right? Well, you would turn this off by, well, you should be able to predict what's going to happen here. Right? Look at this inactive repressor protein. What would happen? This is the allosteric site. This is the active site. So this is the allosteric site. This is the active site. This active site here, can this active site bind to this operator? No, it doesn't have that conformational shape that'll fit that. So tryptophan levels here must be relatively low, right? But if tryptophan levels are high, then I don't need to be making the enzymes that make tryptophan. So I want to shut the expression of those genes down for each of those enzymes. So when tryptophan levels are high, tryptophan probably acts as an allosteric inhibitor here, right? And that is probably what's going to happen. So if tryptophan acts as an allosteric inhibitor, that binds to the allosteric site, and then that will change this inactive repressor into an active one, and then that would bind to the operator there. Right? So I'd recommend going back, looking at the Operon animation. You can also look at this animation here on the PowerPoint, and you should now be able to do this. The trp Operon in this diagram, is it on or is it off? And explain how you know, describe the conditions which would make it so. And then in this situation here, is this trp Operon on? Is it off? Right? Label the parts, the pieces there. Explain how you know if that's on or off. Describe the conditions inside the cell, outside the cell, which would make it so. So just a quick one, right? So if you're confused, go back, look at that at the lac Operon one. Read in your textbook here about this, but you should be able to now, again, describe here the functioning of the trp Operon, and is this trp Operon is said to be a repressible operon, and why? Hint, answering this question here and on the lac Operon one, you have to look at the regulatory gene and the repressor protein there to give you a hint as to why one of these might be repressible, why one of those is inducible then. Okay, so again, asking you finally then to compare and contrast here the lac Operon and the trp Operon, right? Thanks for listening. Bring your questions to class.